Hey MP students, we're back for the Nutrition and Metabolism Lectures, Part 3, coming from Chapter 41 of our patent textbook here at lovely Wayne County Community College. All right, so we've gone over aerobic respiration. Just to quickly review, it had three basic parts: glycolysis, where we netted to ATP and also made some of our reduced coenzyme NADH. And then we ended up with pyruvate as our end carbon product of glycolysis, which will then go on to lose carbon dioxide, and the acetyl group will join up with CoA and enter the citric acid cycle. In the citric acid cycle, which is in the mitochondrial matrix, we ended up netting another 2 ATP, but we made a bunch more coenzymes, 6 NADH and 2 FADH2. These reduced coenzymes are what will go through the electron transport chain. Okay, and then we came out of our citric acid cycle and we took those reduced coenzymes into the mitochondrial inner membrane on those cristae in the mitochondria, and we netted a further 32 ATP. And as I said, that was pretty much theoretical. Studies have found that instead of netting the overall 36 ATP, it's more in the range of like 32, 34 ATP. Okay? All right. So all of the high energy electrons removed during the citric acid cycle then enter this chain of carrier molecules that are embedded on the inner membrane of the mitochondria. And that is the electron transport chain, or the electron transport system. And all these high energy electrons and their accompanying protons are carried to this electron transport system by nicotinamide adenine dinucleotide and flavin adenine dinucleotide. The electrons will then move down the chain from one membrane protein complex to the next, and eventually to their final electron acceptor, which is oxygen, which is always a highly tested question. So remember that. Oxygen is the final electron acceptor. So as these electrons move energetically downhill, the complexes embedded in the membrane of the mitochondrium capture the released energy and use it to pump hydrogen ions from the matrix to the intermembrane space. This pumping forms an electrochemical gradient across the inner mitochondrial membrane, and this gradient is called the proton motive force. The protons can't pass directly through the phospholipid bilayer of the membrane because the core is too hydrophobic. So instead, the protons move down their concentration gradient only with the help of channel proteins that form hydrophilic tunnels across the membrane. In the inner mitochondrial membrane, the hydrogen ions, protons, have just one channel available, a membrane-spanning protein, which is called ATP synthase. And that's what you see there, that big wheel, okay, in that cartoon. So the ATP synthase is a lot like a turbine in a hydroelectric power plant. Instead of being turned by water, it's turned by the flow of the hydrogen ions moving down their electrochemical gradient. And as that ATP synthase turns, it catalyzes the addition of a phosphate to ADP, capturing the energy from the proton gradient as ATP. Okay, so then let's look at this next slide, which is the cell machinery for glucose catabolism. And this is a nice summary picture we've gone through before, where the glucose moves in to the cell and is rapidly phosphorylated to trap it inside of the cell, and glycolysis then occurs in the cytoplasm, the end result being the pyruvate that's made. The pyruvate loses one carbon in the form of carbon dioxide and is now an acetyl group that moves into the mitochondria, and the acetyl-CoA is formed when the acetyl group joins up with the coenzyme A. The citric acid cycle takes place mostly in the mitochondrial matrix, and as it turns around, we make 2 more ATP and some more of our reduced coenzymes. And then The reduced coenzymes that we just discussed move into the electron transport and oxidative phosphorylation, which occur on the inner membrane of the mitochondria. Okay, so that's our little review of what we've learned, and let's now move more. So as we have learned, our first stage of aerobic respiration begins with glycolysis. But some organisms are able to continually convert energy without the presence of oxygen. And even in some human cells, we do that. They will undergo glycolysis, but it's followed by the anaerobic process of fermentation to make ATP. So for instance, muscle cells can continue to produce ATP when oxygen runs low using lactic acid fermentation. However, this will often result in muscle fatigue and pain. And so you'll hear people at the gym complaining of the lactic acid buildup in their muscles. We'll also look at how many yeast will use alcoholic fermentation to produce ethanol. And you can probably guess why it's probably not a good idea for humans to undergo fermentation to produce alcohol. But we'll discuss an interesting case of that too. Okay, so in our introduction to carbohydrate breakdown pathways, we did the whole straight shot down in this cartoon of glycolysis through the TCA and electron transfer, but you can see the little side pathway there over to fermentation, which is what we're discussing now. Both aerobic respiration and fermentation begin with glycolysis. We start out with our 1 molecule of the 6-carbon sugar glucose, and our end result after glycolysis is our 2 molecules of pyruvate. When we go on to aerobic respiration in the mitochondria, we yield a pretty high amount of ATP, about 36 ATP per glucose molecule. Fermentation continues anaerobically in the cytoplasm, and we only yield 2 ATP per glucose molecule. So why would we ever undergo fermentation? Okay, so let's look at that. All right, so sometimes our rate of glycolysis is much higher than the rate of the citric acid cycle, and pyruvate will back up. And in doing this, we get a supply and demand problem from glycolysis, where we end up reducing more of our NAD plus to NADH. And so our supply of the oxidized form of that coenzyme, the NAD plus, becomes diminished, or oxygen just simply isn't available for aerobic respiration. So our purpose of fermentation is that we can continue to regenerate our oxidized form of our coenzyme, the NAD+. So we take our pyruvate and we undergo lactate fermentation. Animals, such as humans, do this. Many bacteria do this. Now, for many plants and yeasts and fungi, they undergo alcoholic fermentation, which we'll also discuss a little bit. And you can see why we wouldn't want to ferment our pyruvate to alcohol. We would be walking around drunk all the time. So let's talk a little bit about fermentation. Okay, so all fermentation takes place in the cytoplasm, and it's done under anaerobic conditions or without oxygen. Rather than oxygen being the final electron acceptor, in this case, the final electron acceptor is an organic molecule. So like aerobic respiration, fermentation begins with glycolysis, but it doesn't fully break down the glucose carbon backbone. So we're not extracting all of the energy out of that carbon backbone. Instead, we're going to remove electrons and hydrogen ions from our reduced coenzyme, NADH, to form NAD+, the oxidized form. And the whole purpose of the fermentation is we want to keep regenerating that NAD+, so we can keep up our glycolysis. Otherwise, it would have to come to a halt because it had no coenzyme available. So our net yield of ATP is the 2 ATP that form in glycolysis. We're not making any more ATP through the process of fermentation. As I said, the whole goal is to keep regenerating the coenzyme. So let's look first at alcoholic fermentation. Alcoholic fermentation takes our glucose and it will convert it to ethyl alcohol. So we undergo glycolysis and form our pyruvate. The pyruvate is then split into acetaldehyde and carbon dioxide. The acetaldehyde will then receive electrons and hydrogen from our reduced coenzyme that we made during glycolysis, the NADH, and will reform our oxidized coenzyme, NAD+, and along with that, ethanol, ethyl alcohol. So this is carried out by yeast, like brewer's yeast, which is used both in bakeries to bake, with all that carbon dioxide being released. It's what raises the bread. And it's also used to make beer, wine, and all these other things that we enjoy. Now, I told you that we don't obviously undergo alcohol fermentation because we would be drunk all the time. And there is an interesting little disease associated with this. And I read an article recently about a man in North Carolina in his 40s who was pulled over by the cops on suspicion of drunk driving. And the police didn't believe him when he said he didn't have any alcohol. And they took him to a hospital and tested him, and his blood alcohol level was found to be 0.2. Legal limit is 0.08, so it was 2.5 times the legal limit, like the equivalent of drinking about 10 drinks or so. And so the man swore up and down that he hadn't had any alcohol to drink. And of course, the cops didn't believe him. The doctors in the hospital, nobody believed him. But eventually, I guess his aunt believed him or something. And it took a while, but she had read some article about a doctor in Ohio who had diagnosed a medical condition called autobrewery syndrome. And so he went to go visit this doctor, and they did indeed find out that he had autobrewery syndrome. And apparently he had had some sort of accident. I think it was his thumb was hurt or something, and they put him on antibiotics, and this changed his gut microbiome. And instead of having all that nice, pleasant bacteria that we all love to grow in our intestines, he grew some fungi, some form of yeast. And whenever he ate a heavy carbohydrate meal, he would have the yeast in his gut digesting the carbohydrates that he ate and fermenting it to alcohol. So he became drunk from the alcohol that the yeast in his gut made. And it took like years for him to eradicate this. And it's quite a situation. And I'll post the article online for you in case you're interested in reading that. But maybe one of you out there will encounter this in a patient someday, because they think it's actually a little bit more common than one would guess. Okay, so let's go on. So alcoholic fermentation, here is just what it looks like. Again, we start with our pyruvate, and when we ferment it, we end up with the first step, forming carbon dioxide and acetaldehyde. Our acetaldehyde then encounters the reduced form of the coenzyme, and we have a redox reaction, and we regenerate our oxidized coenzyme, and we reduce the acetaldehyde to ethyl alcohol. Okay, so that's how you end up with the ethanol. Now, normally for us, we don't undergo alcoholic fermentation. We undergo lactate fermentation. So for our situation, we convert our glucose first to the pyruvate and then to lactate. Okay, so here the electrons and hydrogen ions are transferred directly from the reduced coenzyme NADH to the pyruvate. And in the process, we regenerate the oxidized coenzyme NAD+, and we convert our pyruvate to lactate. In this instance, no carbons are lost as carbon dioxide. So you shouldn't be bubbling out carbon dioxide in your muscle or anything like that. Okay? So lactate fermentation is carried out in our cells under anaerobic condition, but it's also used commercially. If you like yogurt, that is lactate fermentation. When we carry it out in our animal cells, one of the conditions where this is commonly occurring is in our fast twitch or white muscle. So usually during exertion, our muscles will require using a lot of ATP. And once the ATP is consumed and our oxygen content is unable to keep up with the demand, O2 becomes limiting and our NADH starts to accumulate. And so we need to start regenerating some of our oxidized form of the coenzyme. So when the cells need to get rid of this excess NADH, we undergo the lactate fermentation, and pyruvate serves as the electron acceptor, generating lactate and oxidizing the NADH back to NAD+. This is also used in some animals when they hibernate, okay, because they don't have oxygen for long periods of time. So on this little cartoon in here, we have lactate fermentation, and you can see the picture on the right of you have some nice sprinters, and sprinters always have that big, thick thigh muscles, very different from the long distance runners. And like I said, it's that fast twitch muscle that really undergoes the anaerobic lactate fermentation. Okay, if you look over at the left side of this cartoon, you see where we started out with our glucose and glycolysis. We invested our 2 ATP and we made our 4 ATP and came out with pyruvate. And in so doing, in order to make our pyruvate, we had invested 2 oxidized form NAD+, which was then reduced with those 4 electrons to NADH. And you could see that if glycolysis went on and went on and went on, and pyruvate was accumulating, we would also be using up all the NAD+ and generating a bunch of NADH. And if that couldn't undergo aerobic respiration, we need to start regenerating that NAD+ so we can continue with glycolysis. And the way we do that is that second bottom box down there, which is we're using our NADH, and our goal there is to regenerate that NAD+, so we reduce our pyruvate to lactate. Okay? All right, and so this becomes important for understanding what's called the Cori cycle, and that is known as the lactic acid cycle, and it's named after its discoverers, Carl Ferdinand Cori and Gerty Cori. And they won the 1947 Nobel Prize in Physiology and Medicine, not for the Cori cycle, for something else, but let's give them some credit here that they were some very good biochemists or physiologists. So the Cori cycle is the metabolic pathway in which lactate is produced by anaerobic glycolysis in the muscles, and then that lactate moves from the muscles to the liver. And in the liver, it is converted to glucose, which can then return to the muscles. And so it's cyclically metabolized back to lactate. Okay, and here is the cartoon of the Cori cycle. So during muscular activity, the store of ATP needs to constantly be replenished in our muscles. And when the supply of oxygen is sufficient, this energy is going to come from feeding pyruvate, the product of glycolysis, into the citric acid cycle. But when oxygen is insufficient, such as during intense muscle activity, energy has to be released through anaerobic metabolism. And this is where we have the metabolic pathway in which lactate produced by anaerobic glycolysis in our muscles is transported to the liver, and in the liver it is converted to glucose, and that glucose will diffuse back into the blood and return to the muscles, and it's cyclically metabolized back to lactate. The enzyme that converts pyruvate to lactate is called lactate dehydrogenase. And this is a good enzyme to know because you will see it a lot on clinical lab tests. High levels of LDH spilling out in the blood indicates that there is some form of tissue damage somewhere in the body. And we can look at the different forms of isoenzymes to try to localize where that is. It could be liver damage, it could be muscle damage, it could be heart damage. So you'll see high LDH under conditions of a heart attack as well. Okay, so let's now move on to glycogenesis. All right, so glycogenesis is basically the synthesis or genesis of glycogen. This is where glucose molecules are basically knitted together in long chains of glucose to form glycogen for storage. And glycogen is branched. So there will be a branching enzyme that will come in and make glycogen not just as a straight chain, but as a branched chain of glucose. So let's say maybe after a meal, and we have lots of glucose in our blood, and so it starts being taken up by our cells. And once it gets in the cells, it's trapped and phosphorylated and turned into G6P, so it's stuck inside our cells. And all that G6P builds up. But our whole glycolysis and tricyclic acid pathways are sort of overwhelmed. We're producing more, we have more G6P that entered our cell and is trapped in our cell than can possibly go through all that enzymatic process. We basically have a traffic jam of G6P. And just like you, when you're stuck on the highway and there's too many cars, you are going to find an alternate route and get off that highway, right? So what happens with G6P, instead of undergoing the whole glycolysis pathway, it enters the glycogen formation pathway, which is called glycogenesis. Now, when we have high blood sugar levels and that glucose is entering our cells, our pancreatic beta cells will increase insulin in response to that high serum glucose level. And insulin is a hormone that will kind of open up those transporters to let the glucose be taken up in our cells. Glycogen polymers can be made in pretty much any cell, but only two types of cells really store glycogen or make glycogen in any real amount, and that is in the muscle and in the liver. And it is the liver that becomes the reservoir of glycogen for the rest of our body to use it. So when you eat a big meal and your blood glucose gets raised, you have a high serum glucose level. This is your postprandial period. Postprandial means after a meal. Then your blood sugar is raised. And remember, all that blood that's going through your gut, picking up all that food, goes from your gut over to your liver through the portal system. And the liver is where we're going to store all that extra sugar as glycogen. So let's take a look here at glycogen. This is a little branch of glycogen. You have the straight chain at the bottom, and then you have the branch going up at the top. And you can see the picture transmission electron micrograph of the solid glycogen granules in a liver cell. Okay? And then we also need to talk about glycogenolysis, because we make our glycogen, and then when we need sugar, we can break off pieces of glucose from that big giant glycogen storage molecule. Glycogenolysis is essentially a reversal of glycogenesis. Although all cells have the enzymes to break down glycogen to G6P, only a few cell types, the liver, the kidney cortex, and the intestinal mucosa, have the enzyme phosphatase, which allows free glucose to form and possibly leave the cell and travel out through our blood. The liver, which is our systemic reservoir of glycogen, becomes that homeostatic mechanism which helps us maintain our blood glucose level. So in that postprandial period, right after we ate, or in the fed state, insulin will act to cause us to store all that glucose floating around in our blood as glycogen in our liver. But when we're in the non-fed state, hours later, after we've not eaten for a while, let's say you had breakfast at 8 o'clock in the morning and you skip your lunch and it's 1 o'clock in the afternoon, and what's going to happen? You're going to start breaking down your glycogen to get glucose. The pancreatic hormone glucagon acts in the opposite direction of insulin. It is released from the pancreatic alpha cells, and it will stimulate the liver to break down glycogen and release glucose into the blood, hence maintaining that homeostasis of your blood glucose. Glucagon, epinephrine, cortisol, and growth hormone are what are called counter-regulatory hormones, and they act in the opposite manner of insulin. They cause the raising of blood glucose levels. Okay, so let's look at glycogenolysis in a liver cell. Glycogenolysis is the breakdown of glycogen branches through catabolic reactions via the sequential removal of glucose monomers through phosphorylases. This is catalyzed by the enzyme glycogen phosphorylase. The glycogen is cleaved from glycogen to produce glucose 1-phosphate and glycogen less 1 unit of glucose. Phosphoglucomutase then converts glucose 1-phosphate into glucose 6-phosphate, which then enters the glycolytic pathway that you are familiar with. Glycogenolysis takes place in the muscle and liver cells. Here we're looking at the liver cell in response to hormone and neural signals. Now, do you need to know all this? No, you don't need to remember glucose 1-phosphate and glucose 6-phosphate and the names of the enzymes. I'm just telling you so that when you're looking at the slide, you understand it. But what you do need to know is that glycogenolysis is sort of the opposite of what we just did when we had glycogenesis, okay? And this is how our liver is basically going to respond to glucagon, for instance, and it's going to release units of glucose from that glycogen in order to raise our blood sugar, okay? So let's look at the homeostasis of blood glucose levels. And that's our next cartoon here. So this is a pretty busy picture, and I don't like it, but it's in your textbook, so we might as well discuss it. This is about maintaining your normal blood glucose level. So our normal blood glucose level is somewhere around the range of 80 to 90 milligrams per deciliter of blood. Okay, that's your fasting blood glucose level. It'll be higher when you're postprandial, right after a meal. When your blood glucose levels dip below that normal range of 80 to 90 milligrams per deciliter of blood glucose, then your liver cells will increase the rate at which they convert your glycogen into glucose. And conversely, when your blood glucose level increases above this, your liver cells will increase the rate at which they remove glucose molecules from the blood and convert them to glycogen for storage. At even higher levels, say when you look at this cartoon and you see consistent blood sugar levels about 140 milligrams per deciliter, then glucose travels through the blood and goes into forming fat in your adipose tissues. And if you think about it, type 2 diabetics who are always running high blood sugar end up becoming kind of chunky, okay? And at really, really high blood sugar levels, you start excreting it out of your kidneys. And if you have high blood sugars a lot, you actually even damage your kidneys as you're excreting sugar out of your urine all the time. Okay, so let's look a little bit more at the control. If you don't have enough gluco and glyco words to confuse you already, here's another one: gluconeogenesis. Gluconeogenesis is the making of new glucose. Okay, so this is a metabolic pathway that results in making glucose from non-carbohydrate sources. And that's usually being made from amino acids or proteins, primarily that. Less so from fats, but also from fats. Okay, and this process occurs primarily in the liver. And this glucose can diffuse out of the liver and into the blood. In this sense, both gluconeogenesis and glycogenolysis are important for maintaining blood glucose homeostasis by adding glucose to the blood or raising the blood sugar when necessary. And here we see tissue cells and adipose cells and hepatocytes all synthesizing glucose from non-carbohydrate precursors, the process known as gluconeogenesis, and putting glucose or raising our blood glucose levels. And you need to remember that our control of glucose metabolism occurs through hormonal control. Insulin, which is secreted by the beta cells of the pancreas, stimulates the uptake of glucose. Insulin decreases blood glucose concentration by inducing intake of glucose by our cells. Also, insulin activates the enzymes which are responsible for glycogen synthesis. Glucagon, also made by the pancreas, but this time by the alpha cells, will increase the blood glucose level by increasing the activity of the enzyme phosphorylase, which breaks glucose off of glycogen, which is glycogenolysis. Okay, so let's look at the role of insulin. So we have this first picture in A, which starts out with this lovely Big Mac meal that somehow has fruit going along with it, I guess to counteract the fries in the Big Mac. So then you have your high postprandial glucose level, and your pancreas cranks out some insulin from its beta cells, and that will promote the uptake of glucose by the cells of your body, including your liver and your muscle cells, which are well adapted for storing glucose as glycogen. So all that excess glucose is removed from the bloodstream, and then we can look over here at the graph, letter B, and this shows that if the glucose level falls below the normal range, hormones such as glucagon will promote the release of glucose from storage as glycogen back into the bloodstream. Okay, so to further discuss our control, so obviously glucagon and insulin are the two most important hormones you have to remember, and if you're going to remember anything, remember those two. But there are some other hormones that come into play, and we already learned a little bit about the counter-regulatory hormones, also of epinephrine, cortisol, and growth hormone. So hormones that are called incretins will act to increase the amount of insulin released from the pancreatic beta cells and decrease the amount of glucagon released from the pancreatic alpha cells. The incretins will tend to decrease blood glucose levels. Incretins may also reduce the rate of gastric emptying and decrease the desire for more food. So I think I need some incretins. Epinephrine is a hormone that's secreted by the adrenal medulla, and you tend to secrete, it's a kind of a flight or fight hormone. You secrete it in times of emotional or physical stress. Epinephrine increases phosphorylase activity. This makes glycogenolysis occur at a faster rate. So epinephrine accelerates both liver and muscle glycogenolysis, whereas glucagon accelerates only liver glycogenolysis. So say you have a diabetic who's gotten really, really low blood sugar, and they're so low blood sugar that you can't even get them to drink down a glass of orange juice because they're basically getting to the point where they're unconscious. You keep shots of glucagon if you are at a diabetic at home. You keep them in the refrigerator to use in an emergency situation like that. So you can immediately mobilize the glycogen from your liver. Growth hormone is made by the anterior pituitary, and it too will increase your blood glucose level, but by a different mechanism. It causes a shift from carbohydrate to fat catabolism, and it does this by limiting the storage of fat in fat depots. Instead, more fats are mobilized and catabolized, and in this way, growth hormone will spare carbohydrates from being catabolized, and the level of blood glucose in the bloodstream is also increased. Thyroid stimulating hormone, or TSH, is also secreted by the anterior pituitary, and it targets your thyroid and causes it to secrete both T3 and T4, the sort of active forms of thyroid hormone. And these will have complex effects on metabolism. Some of these will raise and some of them will lower the blood glucose level. One of the effects of thyroid hormone is to accelerate catabolism. And because glucose is the body's preferred fuel, the result might be a decrease in the blood glucose level. And we also talked about cortisol, which is a form of glucocorticoid, and this too will increase the blood glucose concentration. Adrenocorticotropic hormone, ACTH, will cause the stimulation and release of cortisol. And since we haven't done the endocrine chapter yet, I'm not going to go into too much of a detail on that right now. We'll discuss it a little bit when we get into the endocrine system. But just know that cortisol is another counter-regulatory hormone, okay? So the opposite effect of insulin. All right. Let's skip ahead a little bit to mechanisms for regulating our food intake. I want to give you a little break from all these anabolic and catabolic pathways, and we'll go on to lipids and proteins a little bit later. Okay, so regulating food intake. Obviously, this is an area that's still not well worked out. If it had been, somebody would be developing the miracle diet pill that works, and we don't have that yet. So it appears that a cluster of neurons in the lateral hypothalamus is the appetite center, meaning that impulses from them brings about increased appetite, or what's called the orexigenic effect, the appetite producing effect. Whereas the ventral medial nucleus area of the hypothalamus is our satiety center, and this is known as the anorexia. And as your textbook notes, what acts directly on both of these feeding centers to stimulate or depress our appetite is still being worked out. But we know that one factor is the temperature of blood circulating to the hypothalamus. A moderate decrease in the blood temperature will stimulate the appetite center and inhibit the satiety center. So the result is the individual has an appetite and wants to eat. An increase in the blood temperature produces the opposite effect, a depressed appetite or anorexia. One well-known instance of this effect is the loss of appetite in people who have a fever. So right now, when we're hearing about all these folks who have COVID-19, the big primary symptom that's an ominous sign for them is being febrile. And I'm sure that they're having a great lack of appetite when they have the COVID-19 fever. Researchers have also identified a number of hormones that have an effect on regulating our food intake. One of these is the ghrelin gremlin, the gremlin that causes you to be hungry. It's a circulating hormone produced by the enteroendocrine cells of the gastrointestinal tract, especially in the stomach, and it is called the hunger hormone. Ghrelin is the gremlin that causes you to increase your food intake. See, I say it again, and you won't forget it on a test. Leptin is basically the opposite of that. It's a hormone predominantly made by adipose cells and enterocytes in the small intestine that helps to regulate energy balance by inhibiting hunger. So that in turn diminishes fat storage in the adipose sites. So the importance of proper nutrition begins at the moment of conception and continues throughout our life. It is critical for the fetus to get the proper nutrition and vitamins. This is why we put so many women on prenatal vitamins. We need to have things like folate for proper neural tube formation. Protein and calcium are critical in fetal development, infancy, and childhood. Protein is a critical nutrient during fetal development, infancy, and childhood. Sufficient proteins that contain all the essential amino acids are required to permit normal development of the nervous system, muscle tissues, and other vital structures. Sufficient calcium is needed for the proper development of the skeleton and other tissues. And unless a pregnant woman consumes enough calcium, it'll probably go to the baby first, and then she might experiencing the bone softening effects of calcium deficiency. In fact, a long time ago, I read a study where they examined the skeletons of Eskimo women who had lots of babies and breastfed for a long while, and they all had very, very early osteoporosis. In late adulthood, the number of food calories that we need declines because we have a declining metabolic rate. The metabolic rate decline is thought to result largely from age-related changes in the balance of metabolic hormones such as T3 and T4, the thyroid hormones. Even though the number of required food calories declines, the overall balance of nutrients consumed must be maintained to preserve our proper metabolic function. Okay, so let's look at protein calorie malnutrition, because that's usually highly tested. And you see the pictures here that I unloaded off the internet. And kwashiorkor is a protein deficiency under conditions of having adequate energy intake. So in kwashiorkor, they're having normal calories. It's just protein in particular is deficient. And so you see the child here with a large belly. They get peripheral edema, and they get this reduced subcutaneous fat, but a big fatty liver. They'll have things like dermatitis and sort of a flaky paint appearance of the skin. And this has a worse prognosis than the other sort of starvation, or which is marasmus. So in marasmus, you have severe deficiency of all the nutrients. And so you have generalized inadequate caloric intake. They don't have the peripheral edema. Their skin becomes kind of dry and wrinkled, but they don't have a dermatosis sort of rash. These kids tend to have a voracious appetite, where those with kwashiorkor actually end up having a poor appetite. And they don't have the fatty liver. They're just generally starved and skinny looking. Okay, and they don't have a fatty liver, and you have a better prognosis. You have a better prognosis of refeeding these children. Okay, and it's not always a clear-cut picture. As you can see, you can have this combination of marasmic kwashiorkor. So sometimes both things can kind of coexist. Cachexia is what is referred to as a wasting syndrome, and you tend to see this especially in HIV or some cancer patients, other chronic conditions. And so this is extreme weight loss, particularly with loss of skeletal muscle mass. All right. So let's talk a little bit about the body mass index. The BMI is a measure of your body weight compared to your height. And so it was developed as a way for researchers to assess body fat percentages in large populations, not so much to keep track of an individual, although quite frankly, we've adopted it and we use it for individuals in clinical practice because we don't really have anything better. And it's simple enough. You punch in a patient's weight and their height into your electronic medical record, and it spits out your BMI usually automatically now. And but it can fool you a little bit. So like really elite athletes, especially heavier, dense bodybuilders, professional athletes like football players, will often have a BMI that puts them in the obese category, but they are anything but obese. I remember Sports Illustrated had a big thing where they showed a lot of the athletes, and they had them in these kind of fancy photographs and semi-nude, and they showed their BMIs. And so you'd see an elite athlete, a great sprinter like Jackie Joyner-Kersee, and she has those big, powerful thighs, and she has very low body fat. But her BMI was high because she had really dense, heavy muscle mass to do the amazing feats that she did. So knowing your body mass index, we all should know where our normal body mass BMI is in the 18.5 to 24.9 range. So you're overweight if you're between basically 25 and 30. You're underweight when you're below 18.5. And the severely underweight, starving, anorexic type people are below 16.5. But again, not always so. There are some naturally thin people who are not anorexic. A BMI over 30 is when equal to or over 30 is when you're considered obese. And then we rank obesity by class. And they do this for also considering those who get bariatric surgery. Usually if you're between 30 and 35, you have to have either one or two obesity-related conditions such as type 2 diabetes and hypertension, and then you can get your insurance to pay for bariatric surgery. I think that if you're obesity class 3, over 40, they make you lose weight and get down into the 35 to 40 range before they actually will do the bariatric surgery. It's very fascinating to deal with all that. I have a friend who is a bariatric surgeon. Okay, so let's look at some nutritional disorders. Metabolic syndrome, also known as Syndrome X or insulin resistance. This is a multifactorial disease with multiple risk factors that arise from insulin resistance that accompanies abnormal adipose deposition and function. It comprises a combination of risk factors for coronary heart disease, as well as for diabetes, fatty liver, several cancers, stroke, and polycystic ovarian. This is a common cluster of symptoms and is pretty much at a national crisis level. So let's go over some of the signs and symptoms. Clinical manifestations of metabolic syndrome can include hypertension, hyperglycemia, insulin resistance, abdominal obesity or excess body fat around the waist in particular, abnormal cholesterol and triglyceride levels, acanthosis nigricans, which is a velvety hyperpigmentation of the skin that's usually found in, and a lot of this can actually be reversed by getting control of the diet. And for metabolic syndrome in particular, the ketogenic diet actually works pretty well. So if you know people in your family who are having issues with this, tell them to give the ketogenic diet a try. It might actually work. Okay, the eating disorders, anorexia nervosa and bulimia nervosa, primarily used to be thought of as diseases of young adults, primarily women, but now we're finding these diseases linger on into middle age. These disorders are interesting because they don't have just a psychological component, they also have a societal component. I remember back in 1999 at the American Psychiatric Association meetings in Washington, there was a presentation, and they talked about how in French Polynesia, like Fiji, that as soon as they introduced American TV there, all these women who used to enjoy having a sort of robust, nicely rounded body, which was the norm in their culture. If you ever looked at Paul Gauguin's paintings from French Polynesia, you'd know this. And as soon as they introduced American TV, they had American problems. And all the young girls dreamed of looking like American super thin movie stars, and they had the introduction. So anorexia is an eating disorder that's characterized by weight loss and inability to basically maintain a normal BMI. So they have difficulty maintaining appropriate body weight for height, age, and stature, and in many individuals they have a distorted body image, body dysmorphia. People with anorexia generally restrict the number of calories and the types of food they eat. They can also over-exercise. You'll see the, say girls, because it's primarily girls, on the eating disorder floor in the psych wards. I was at UNC, which has one of the biggest eating disorder clinics in the nation, so I got to see this. And you know, they'll find the strangest ways to exercise. They'll do sit-ups in their room all night long. It's a really interesting disorder. Bulimia is an illness that's characterized more by eating, vomiting, or purging cycles. And so these people can be basically normal weight or even a little bit overweight. And it tends to affect about 1% of college age students, and the mean age is about age 25. People with bulimia have an uncontrollable urge to eat food that leads to massive overeating or binging, which then is followed by the purging cycles of either vomiting or abusing laxatives. And so they can often end up with a lot of distorted lab values because of the alternating binging and purging cycles. These electrolyte imbalances they get can lead to the development of neurological problems, things like convulsions, tetany, seizures. Because they're vomiting so much, they can get things like aspiration pneumonia. And you see a lot of dentists will be, or dental hygienists will be the first ones to recognize this because they'll see erosion of the enamel on their teeth from the continued vomiting. You can look at their hands because they put their fingers down their throat to induce the vomiting, and they'll have scarred from the teeth marks all over the backs of their hands and fingers. There's some interesting associations with bulimia. I recall that a lot of people with bulimia have been sexually abused and have chemical dependencies. So a lot of interesting issues go along with these eating disorders. And I told you before that to try to make their eating disorders socially acceptable, you can find in teenage girls who suddenly decide, I'm becoming vegetarian, or I'm not going to eat such and such food group. And you think, oh, they're going through a phase. Well, the phase might actually be an eating disorder. So that should ring true to you when you see this amongst teenage girls, because this is usually when these things come on. And so a sudden change in their dietary habits should be a big red flag. And this leads me into discussing a newer eating disorder. This one's not in your textbook. And this is called orthorexia. Orthorexia is a term for a condition that includes symptoms of obsessive behavior in pursuit of a healthy diet. Those with orthorexia often display signs and symptoms of anxiety disorders that frequently co-occur with anorexia or other eating disorders. And there's probably a lot of crossover amongst all of these. A person with orthorexia is obsessed with defining and maintaining the perfect diet rather than the ideal weight, like an anorexic. This person will fixate on eating foods that give a feeling of being pure and healthy. An orthorexic might avoid numerous food groups, such as those made with artificial colors, flavors, or preservatives, pesticides or non-GMO, fat, sugar, salt, gluten. Gluten's a big one. Animal or dairy products, and avoiding gluten when you don't have celiac disease. There's a whole variety of ingredients they'll avoid that they consider unhealthy. They have common behavior changes that might be signs of orthorexia, such as obsessive concern over the relationship between their food choices and health concerns with asthma, digestive problems, low mood, anxiety, or allergies. They attribute everything to the food they're eating. They have increasing avoidance of foods because of food allergies that might not even exist without, having medical advice about them. They have a noticeable increase in the consumption of special supplements, herbal remedies, and probiotics. A lot of these people also work in things like the fitness industry. You'll find a lot of anorexia amongst dancers, for instance. A lot of fitness instructors will be orthorexic. So these things kind of go together, and they should all be red flags for you, okay? And of course, we have the number one eating disorder in America, which is obesity. It is a public health crisis. Our adults and our children are obese. And it is far getting to the point where it's surpassing cigarettes as our number one. Okay, so let's get to some practice questions, all right? All right, number one. Which of the following types of cells contain phosphatase? So think about what phosphatase does. And you can kind of look at these and think one of these is not like the others. So again, pause, think about it, and we'll move on to the answer. So it's the renal cortex cells, the kidney, that contain phosphatase. Because remember, it's in the kidney and the intestinal mucosal cells that you can shunt the glucose once it's been phosphorylated. You can reverse that with the phosphatase, and it will go right back out the glucose transporter. The sequential removal of glucose. And the answer is glycogenolysis. Glycogenolysis is breaking off the monomer of glucose from the glycogen. Okay. Which of the following hormones does not raise blood sugar levels? Okay, so there is one definite here that is not like the others, and that would be insulin. And I see I must have slides out of order because that's not coming up as the next slide. So insulin does not raise blood sugar levels. It lowers it and forces the glucose to go inside of the cell. Okay, so Christy works in the fitness industry, and she suffered from bouts of postprandial intestinal discomfort, which is in her words. After consulting Google, Livestrong, and various websites, she has completely eliminated gluten, meat, and dairy products from her diet. As well, she only buys organic produce. She recently started taking probiotics recommended by the teenage clerk at GNC, as opposed to somebody who actually has studied something about this. She most likely suffers from which of the following? Anorexia, orthorexia, bulimia, binge eating, or rumination. And I think you know the answer to that because we just went through it ad nauseum, right? So the answer is orthorexia nervosa. Okay, but there is a lot of crossover between all of these. And here's a slide that's out of order that shows you indeed it is insulin that does not raise blood sugar levels. Sorry about that being out of order. Okay, gluconeogenesis is the process of conversion of glucose to pyruvate. See, there's another typo there. Sorry about that too. So gluconeogenesis is the process of conversion of glucose to pyruvate. And this is false, okay? This is false because it is the opposite of that. Gluconeogenesis is the conversion of pyruvate to glucose. All right. Which BMI would most likely be the BMI for an actively anorectic teenage girl? Okay, so she's actively anorectic. She's not recovered from this. Okay, that BMI would be 17, so it's below 18.5, right? All right, and where is the appetite center located? Okay, where is the appetite center located? So this is stimulating the appetite. And this is in the hypothalamus, specifically in the lateral hypothalamus, right? All right. Which of the following conditions is caused by chronic protein deficiency? Chronic protein deficiency. Okay, and that is kwashiorkor. So remember, kwashiorkor is specifically protein deficiency, not overall calorie deficiency. Marasmus is overall calorie deficiency and not protein in the balance of our nutrients. Okay? And cachexia is when we have muscle wasting. Pellagra and beriberi are vitamin deficiency disorders that we haven't gotten to yet. All right, the action of phosphatase enzyme is to catalyze which of the following? Okay. Does it unlock the glucose transporter? Does it phosphorylate glucose? Does it mobilize the glucose reserves from glycogen deposits in the muscles and liver? Or does it add branches to the growing glycogen molecule during the synthesis of glycogen? And I hope you picked that it mobilizes the glucose reserves from glycogen deposits in the muscle and liver. All right. The purpose of fermentation in yeast is the same as that in muscle and bacteria, which is to replenish the supply of NAD+ for glycolysis. And that is true. The whole point of fermentation is that it doesn't make more ATP, but it does replenish our coenzyme. It does replenish the oxidized form, NAD+, for glycolysis so that it can continue to proceed. Okay? All right, and the next time we meet, it will be our last from chapter 41. It's a big chapter. Usually everybody always likes the digestive system, and it's a little bit unusual that all this metabolism and the Cori cycle and all that is in with all the digestive chapter things. Usually they're separate biochemistry oriented chapters. But I hope we've made it a little bit less miserable than it needs to be. Okay? See you next time.